Welcome in everyone to Tofu Tech Talk. Camera is just coming in a little bit delayed. There we go. We're here. Tofu Tech Talk is, as you guys know, a special podcast that I do about I say once a month. It's been once a week, quite consistently, where I invite Twitch streamers with STEM backgrounds. And today we actually have a really, really special guest. I think this might be one of my first ever people who found my stream, we'll say, with STEM backgrounds, because you used to be here back in the days when I was just
hmm
streaming a little bit of our variety gaming years ago. So, anyways, without further ado, AriellaStudies, please introduce yourself as much or as little as you would like.
Thank you so much. I'm really excited to be here. You're actually one of the first people I ever followed on Twitch. I've only had a Twitch account for like a year. So I think I followed you before I ever even started streaming.
Wow.
So maybe you're a little bit of inspiration there. Yeah, we actually just had my streamiversary. My very first stream was April 26th. I wasn't able to do a party or anything because I'm just getting over COVID. I had COVID during it. But anyway, introducing myself. I'll try to stay on track. So, I'm Ariella. I'm an MD PhD student. So what that means is it's a combined medical degree and a PhD doctorate like Tofu has. Instead of doing something logical like MD and then PhD or PhD and then MD, what they do is if each of these fingers is like a year in the program, they take the MD and they just like make a little chunk in the middle and then just shove a PhD in there. So I've already done the first half of med school and I'm in my last year of my PhD and then I have to go back for the remainder of med school after that. I always find the fingers help explain it to people. So it's really long.
No, that makes sense.
Yeah.
So, how many years are you now into your both MD, PhD program?
7 of 9. Cute little Star Trek
Do you have a
reference there.
master's as well?
No,
So,
no. I
bachelor's.
decided, yeah,
Okay.
bachelor's of science going into it. So yeah, going a little farther back in my background, I graduated. I went to Berkeley, go Bears. Studied molecular cell biology and Japanese. Um, then graduated, moved to Japan for 4 years, kind of randomly, and just worked there, doing kind of typical what a pre-med would do. Like I worked in an academic research lab. I just did it in Japan instead. And then I came back to America to apply to medical school because the system is totally different. So I wasn't going to do it there. And started in this program. And the program takes forever. So yeah, 7 of 9,
It's
seven of
very
nine
good.
years. So I call myself a forever student, and my name is AriellaStudies because I just feel like I've been studying forever and will be studying forever. It never ends.
Now that is incredible. And is that normal, this program? I've never heard of this, doing an MD and a PhD. Like, why? Why would you do that?
Why? That's a great question. That's a question I asked myself very frequently these last couple years. It seemed like a great idea when I was 26 and didn't know exactly what I was going to do with my life, and I was like, "Oh, an MD and a PhD? I don't know which one to do. So por qué no los dos? Why not both? That's an option. Yay." I have some regrets sometimes. Um but yeah, it's a real thing. I didn't make it up. I guess if that's what you mean by normal, it's a program. Typically, uh, the big programs are sponsored by the United States government, by the NIH. So the idea is to fund these programs to create more medical scientists. So they want people with medical degrees who also have a strong scientific background. So it's called the Medical Scientist Training Program, MSTP. And the big advantage of it is, well, you obviously get both degrees, but also you don't have to pay for any of it. They give you a stipend for the whole 9 years,
Nice.
which is a big deal in America because medical school here is extremely expensive. So I don't have to pay for a dime of it. So I'll graduate with my medical degree without an ounce of student debt. And most people can't do that. So that's the idea is, oh, well, we're going to take like a decade of your life from you, but at least you don't have any debt. So that's the trade-off.
Man, that is, I've had so many American guests here, so it's such a heartbreak to hear all of these problems with college debt and university debt. It's always so heartbreaking to
Yeah.
hear. But yeah. Okay, so you've committed to 9 years of an MD and a PhD, and you're just finishing your MD. So you're finishing your PhD now. Will you
Yes.
have two separate award ceremonies?
Yes. So, well, it's a good question. We don't really get a big hullabaloo with the PhD, especially because we can finish it at all different times. So I don't think I'm going to any kind of graduation
Okay.
ceremony. I just have my thesis defense day, which I'm trying to schedule right now. It's like
Nice.
herding cats. Like I need to get 5 professors in the same room at the
Oh,
same time,
five.
and they keep changing their freaking schedules on me. Like I already sent out the calendar invite and they're like, "Oh, actually, I'd like to fly back from DC a day later. So can you start over?" I just got that email yesterday and I'm
Oh,
like,
Jesus.
"Ah." Because I really need this date. I'm like, "I need to graduate." And yeah, so so we have the thesis defense. I'm not sure if it's the same way that it works in Ireland, but yeah, I get 5 professors in the room. They're my committee. I give my presentation about my research. In ye olden days, they used to be able to like reject you and have you go back to do more PhD. That doesn't really happen anymore. It's more of like a ceremony now. Like they've already approved, you've already sent in your dissertation and they've given you the green light
Absolutely. Yeah.
beforehand. So you have your family fly in. It'd be super awkward to have like your parents and everyone come in and they're like, just kidding, rejected. So it's more so it's more of just like a celebration. So that is theoretically taking place at the end of July, as soon as I get these people to agree on a date. And I forget the rest of the question. Then I go back to medical school.
No worries, no worries.
And then graduation. Yes.
Yes.
Then I get a real graduation at the end of medical school 2
Okay.
years from now. Yeah.
Okay. That's perfect. No, the defense sounds very similar. You said 5 professors. I guess I
Yeah.
did have 5 professors in my room, but I actually, the Irish one is a bit different in the sense that we have only 2 technical examiners. One examiner is your internal. It's a lecturer or professor from the university you're getting your PhD in, and then you have the external. Mine was from the United States as well.
Oh, wow.
Flew in, but again, it was a connection through the society, and usually the idea behind it is you get someone who's not an expert in your field and someone who is an expert. You want 2 of those, and then obviously we have the chair and these other people, but they're not officially examining you. So does that sound similar to the American system?
I haven't actually done my defense yet, but
I'll let you know.
I'm not having no one's flying in. I pretty much picked everyone on it, which is nice. Because I can purposely not pick any assholes.
No, absolutely.
Just
Yeah.
yeah. And yeah, so I I so I haven't actually done. So I've watched other people's defenses, but only the public part.
Yeah.
So they do a presentation and then they kick everyone out, and
Yep.
then it's just the student and the committee left. And so I've never actually seen that. So I don't really know exactly how it's gonna happen, but I assume they'll ask me some questions, and hopefully I'll be able to answer them. And yeah, I'll have to get back to you in like 2 months on how it went.
If it's anything like mine was last year, and my only advice is that like don't take it, but like you'll be over prepared anyway. You're going to prepare for your defense. The questions are going to be way nicer than they actually. You're going to prepare for the worst, and the worst will not happen. They're a lot nicer, and and you're right about it just being a ceremony rather than because they've read your thesis, and basically if it wasn't a good enough thesis they would have contacted the supervisor and said, look, this is not a good enough thesis for a PhD. So obviously you've qualified. They just want to make sure that you did the experiments, that you know what's going on, and I think that's it. But that's fantastic to hear that you're finishing your PhD, and this is actually what we want to talk about. What is your field of research? What is your PhD about?
Yeah, so I study the genetics of aging, and I actually have a little pitch to try to get people interested in this before I dive into the nitty gritty. So, one thing that I like to bring up is All right, so we know that we have the typical laboratory rodents are, mice and rats. I know you guys are really into your lab rats here.
Oh, I
Um,
wonder why. Hmm.
but there's also, other members of the rodent family are squirrels, naked mole rats,
Bunnies?
and it, maybe, I don't know,
Okay.
I think so. But anyway, they're what's really interesting is that even under like very safe laboratory conditions, mice only live to be a maximum of like 3 or 4 years, similar with rats. Squirrels, which are pretty much the same thing, they just have those fluffy butts, can live to be about 20 years.
Oh, they're
So,
like
five
chinchillas.
times.
Oh,
Yeah, so like
wow.
5 times, even though they're pretty much like the same size, pretty much the same physiology, at a macro level going on. And then you get to naked mole rats, and naked mole rats are very, very interesting. They can live to be about 30. But also they don't really age in the traditional sense. They say they experience like negligible senescence is how I would put it. And the way you can think about it is, so you know how when you're a human growing up and you're kind of like 25, you assume like probably not going to die today. Like I'm 25, like unless something crazy happens. But if you're like 95, you're thinking like, all right, any day now, the likelihood that I'm going to die of something has dramatically gone up by
Hmm.
then, right? It's like exponentially increases with age. Naked mole rats aren't immortal, they're not gods, but their probability of death on any given day is pretty much the same. Like they don't experience an increased probability of death when they get older. So they're very, very special. And so my pitch to you is, these are at a macro level pretty much the same tiny little mammal running around on the ground. What is actually going on that makes them age in such different ways? And can we figure out what that is? And can we figure out on a scale of mouse to naked mole rat, like for our size and who we are humans like the mouse of our size or the naked mole rats of our size or the
Ah.
squirrel of our size, and our physiology. And so like if we're like the rats of our realm, is there something that we could do to upgrade us to be like the squirrels or the naked mole rats of our realm? Because that would be a huge step up.
Hmm.
So that's one of the things that got me really interested in the genetics of aging. Did that help you be more interested in the genetics of aging?
Yeah, but now we're interested in immortality. Give us the recipe so that we can
Yeah.
live long and, live to an age where we can
Yeah.
go to the lawn and chase kids off our lawns. We need to know
Yeah.
this.
I saw in chat someone asking why do they die at 30 then. I think it's just so I'm not a naked mole rat expert. But like I said, they're not immortal. So if you have, X% chance of dying on any given day and you live enough days, by the time you get to a certain age, you'll have run out your chances. Like it'll come for you
Is this,
eventually.
yeah, it's just pure statistical probability then.
Yeah, like you'll get an infection or something, and like they do get cancer much less than other rodents, but it is possible to get. So it's like all these different things that could happen, like an accident, an infection, an intestinal blockage, a rogue
Hmm.
cancer starting, even though they're all pretty unlikely. By the time you've added up all the times they could happen over 30 years, they pretty much something has happened by then.
Hmm.
And they don't have like I don't know. Like we don't have like a naked mole rat ICU. So, when something happens, they just kind of die. Yeah.
Okay, so naked mole rats are the recipe, the backbone recipe for immortality. Is the basis of your research? Is that a fair statement or not?
I mean, so it's not what I directly study. It's just something that I find is really easy for other people to understand and get excited about the realm of the big umbrella of aging research in general. There are people there are aging labs that study naked mole rats. We don't have any at WashU. If we did, I would have been all over that. I visited one in Japan and it was the cutest thing. They had like little naked mole rat plushies everywhere, of course. They had their own little naked mole rat like mascot, and the lab system was so cool. It was, you know how the hamsters, when you see like a hamster enclosure, it's like all the little tubes that go everywhere. Like they
Yeah.
had that like all over the lab. So it wasn't like with mouse labs where they just have their own, you know, little isolated cages. Like they had this big glass enclosure with all kinds of tubes, and you could see them because they're burrowing creatures. So there's
Yes,
like almost
yes.
like a two-way mirror so that we could see what they were doing.
Mm.
And oh, I would love to work in a lab like that. And they
And this
seemed
is
so
Japan.
happy. This is Japan, and they
Mm.
had so much room to run around. I don't personally work with animals. Um, like my options would have been mice, and I actually don't agree with the standards we have for working with mice and rats, at least in places that I've been. So I don't actually do animal research. But well, actually, okay, technically I do animal research, but it's microscopic animals. It's
Ah,
microscopic little nematode worms. Um,
okay. And we
yeah,
had
so
science last, well, we had Volcano Doc before, and before that science, and
yeah.
you were here, and he told us that apparently, according to animal ethics, anything like bugs, ants, worms, they're not classified as an animal, so they're not under protective rights.
Exactly. Yeah. So yeah, invertebrates, you know, like the worms that I have, there we don't have to submit any approvals to the UCAC. I can't remember even the acronym because I never have to talk to them. There's a whole board, and you know you write up what you want to do and submit it, and they have to approve it, and
Mm.
there are veterinarians that watch over them. Okay, it's not like we throw them to the wolves. Like there are standards. I just personally the standards aren't up to what I would want them to be, so I decline to participate in that. Like for primate research, I think the standards are excellent. So people have a hard time understanding my position sometimes. Because if I was invited to a primate research lab, I would say yes. And they're like, well, how could you possibly be okay working with primates, but then you won't experiment on mice? That makes no sense. But you have to realize that my position is a little bit nuanced, and I would agree to work with primates because the standards for what you for how you have to treat them are so high that I
Mm.
agree with those standards. And the standards for mice, while a lot of people agree with them, I personally don't. They're much lower than primates for good reason, but I think they're too low. So that's why I don't participate in those labs.
Okay, so the lab rats in the chat, you're safe for now.
Yeah.
You're
Oh,
safe for now.
my partner is in chat giving me the acronym because he does work with mice.
Nice.
IACUC. Yeah,
I
IACUC. IACUC. Yeah, that's the that's the people that we have to ask.
Perfect. So before I get sidetracked, because we have a few questions already, so chat,
Yeah.
make sure if you guys want me to log any questions that you use the channel points. That way I keep track of them, and I will go through them. But I'm more important than you guys, so I get to ask my questions first. So you're researching on this theory of age. Can I because I don't know if you have an embargo on your thesis, if you haven't got your publications yet. Tell me where you can't answer the questions. What
Yeah, I could.
exactly do you do
Oh.
in the lab? Sorry, that's kind of the question I was going. Like, what do you
Mhm.
do in the lab? How much can you tell me about your results of your research so far? Any conclusions from it?
Yeah, I could pretty much tell you everything except that I would, just to err on the side of caution, I won't tell you the specific genes that I found. That's like the more protected information, but I can tell you my methods and like the broad patterns of
Yeah,
my results. I think I don't think there's anything wrong with that, and I've already presented this at numerous conferences, so it's not like a
secret.
top secret confidential government embargo or anything. So yeah, what I do in lab, let me see where I want to start. So I study these teeny tiny worms, and the reason I picked them, besides the fact that I don't want to hurt anything with a fuzzy little face, they are actually an excellent model for aging.
Oh yes, you made a
Yeah.
joke about this during science stream.
Yeah, yeah, they're the best. Little worms, C. elegans, they're the best. They've been really popular for decades for aging research because they experience aging and senescence in a similar way to humans, as similar as you can get for microscopic worms. So they actually when they get older, they get all wrinkly. They get sarcopenia, which means like their muscles lose strength, so they're like wrinkly, they're frail. They experience what's called post-reproductive decline. So like after they're done having babies, everything sort of starts to get frail and fail in a curve that's very similar to the human curve, just on a much smaller scale. Whereas naked mole rats, for instance, don't experience that at all. So they'd be a terrible model for human aging research. And another advantage of worms is they only live for a few weeks. So if I wanted to do lifespan extension experiments in mice, even if I started that experiment on day 1 of my PhD, I'd be there for easily 5 years just with that one experiment, you know? And it's not practical for a PhD. I mean, you can buy mice that are like pre-aged. There are ways to get around it, but it is definitely a lot harder. You can definitely test a lot fewer things because it takes years as opposed to just a few weeks. So that's why I use worms. And my like elevator pitch of my research, I'll try to keep it tight so you can ask follow-up questions, is basically, so we're interested in the aging process. Like we said, we want to know, are we the mice, are we the squirrels, or are we the naked mole rats of our realm? We're definitely not the naked mole rats. I shouldn't have even said that. The mice, the rats, the squirrels. And there's a lot of different ways to study that. One way that people are really interested is in looking at differentially expressed genes. And that's just sort of fancy words for what genes are being used more or less with age. So we take tissue samples from young beings and young adult beings and old adult beings and compare them and see which genes are turned on more in the old or turned on more in the young, and then we can kind of see what things are really changing at a genetic level with age. And there have been tons of studies that have done that, so there's all this data already banked, so I don't have to go and get my own tissue samples from all the mammals. This stuff has already been banked, but people have trouble figuring out what to do with it. Because when you run those experiments, you end up with a list of, maybe a couple hundred genes, and it's like, all right, these are really different with age. Here's 200 genes. And then you're like, what now? And a lot of people's what now is they'll try to find a pattern. There are some standard statistical computational methods of figuring out, oh, well, what are the patterns? Can we do gene ontology analysis? And it'll say really shocking things like, oh, the immune system changes with age, or like, oh, your connective tissue starts to decline with age. And it's like, no shit, Sherlock, we knew that already. And so you just kind of get these patterns, and they're mostly sanity checks. It's like, all right, you clearly did your analysis correctly because all of this makes sense, but it doesn't actually push the science forward that much. And you can't just take those 200 genes and throw them into mice because it would cost millions of dollars and take 1 billion years. So my idea was, let's do a big meta-analysis of 25 of those previous studies. 25 is an arbitrary number. I just didn't want to do 100. Ain't nobody got time for that. And like 25 of those previous studies, comparing young versus old, old tissues, get all that data, crunch it up, get a list of maybe 50 candidates that show up the most frequently across those 25 to kind of root out the noise, because aging research, it's not like a really hardcore disease. The signals are like very subtle, so it's hard to pick up if you don't do a meta-analysis, a big analysis with a lot of different data sets. So I did my meta-analysis. I found 50 that were showing up all over the place, human brain, mouse kidney, just showing up everywhere, right? And then, but my that wasn't my big idea. That was just the preliminary thing. That was just the starting point to get the candidates. My big idea is, let's plug those into worms. We have the time. It's hella fast, hella cheap. We can't do it in mice. We could do it in worms. And another fun thing about worms is actually over half of human genes have a pretty reasonable ortholog or equivalent in worms. So you really can test what a lot of human genes would do to lifespan in worms. We have like half of them in common, even though they're like smaller than a grain of rice and all they do is wiggle and scoot, you know? So that's what I did. So out of the 50 candidates, about 25, 30 of them had a reasonable ortholog and equivalent in worms. I had to throw out a few other ones because just for practical lab reasons that come up, I couldn't make the chemicals that I needed to affect to impact those genes effectively for whatever reason. The reagent wasn't commercially available, just dumb reasons. So narrowed it down to like 20, and I tested those in worms, and I got back originally I got back like 10 that significantly extended lifespan. But reproducibility is really important, and wonky things can happen when you only do an experiment once. So I did it all over again for all the ones that had shown up as a positive. Reproducibility is super hot right now. And then so I narrowed it down to 6. So there were 6 genes that significantly and reproducibly extended lifespan in worms. And I think that now I'm having fun. Now I'm having fun researching those genes and, hopefully writing a really cool discussion section about why I think those came up and if any of them are, worth taking forward to look into for interventions in, extending mammalian lifespan, or just learning something cool academic from it. So does that make sense? Was that too long? Sorry, I tried to make it an elevator pitch.
No, I thought it was really, really good. So the summary, if you want me to summarize, so I understood everything, is I suppose, where do I start? You got already all the analysis that was already there. You selected the genes that had the relative corresponding genes to the human genes, and you performed your tests with reproducibility. You showed that 6 genes clearly did prolong the lives of the worms, and yeah. I guess the question kind of leads on from there to say, what's the next step? Is it to put it in mice? Is it to put it in something a little bit more, or do you think you need more reproducibility, like other labs, for example, other lab practices?
Yeah. So I think I see my study as a pilot study, and we can talk more about this if you want, but my PhD has been a bit of a dumpster fire. So I've now outlasted 2 PIs. The PI is principal investigator, like the boss of the lab. So my first PhD lab shut down, my second PhD lab shut down. So it has been a crazy road.
That's
so
rough.
basically what I've ended up doing is my PhD, like, it's the most like DIY, MacGyver, cheap ass, like all on my own PhD ever. Like there's no lab doing this. It's just me. And like there's no big R01 grant funding me. Like I paid my own salary with a T32 grant for a while. Right. It was wild. I had to go nuts. Like I had a formal complaint letter. I wrote it up. It had a table of contents. It was 14 pages. I sent it to the dean of the entire like umbrella program of the university. I had meetings and I was like, listen, y'all got 2 choices, okay? You get me this equipment or you have to kick me out because that's the only way I'm finishing my PhD. And MSTPs, okay, not to like sound a little bit arrogant, but in the context of like a medical graduate school, the medical scientist training program people, we're kind of important. Like they don't want to piss off the med school by like interfering with our PhD. So we actually have a little bit of pull. So I was kind of like, you'll have to kick me out and like you'll have to answer to the MSTP office if you don't get me this equipment. They're like, all right, we're gonna get you the equipment. Like, calm down. Because I made it clear. I was like, I'm not letting up. Like, I sent emails to this dean. I sent emails to that dean. I will go up to the vice provost. Like, I am annoying and stubborn enough. Like, I will make this your problem if you don't think it's your problem. And they gave me what I needed.
Perfect. And you were able to get then I was trying to remember what the original question was.
I'm so sorry. My bad.
No, I forgot too.
Oh yeah, it's a pilot study. So you were
Okay, there
saying
we
what's
go.
the next
Yes.
step?
Yeah, next step. Yes.
So so I don't have like resources, money, staff. I don't even have a lab tech. So I'm not trying to do like the best experiment that's ever going to blow your socks off at the hugest scale. I'm pitching it as like a small pilot study that's a proof of principle. And then, my discussion and future direction section of my paper is going to be like, I would be so thrilled if other labs, because I'm done with this shit, if other labs, not me, take these principles and apply them using like fancier shit, like large scale RNA-seq, maybe you can do 100 data sets, like get even better quality data. Maybe you can do, 3 experiments instead of just 2, and you can have 2 different humans scoring it to remove some human error and subjectivity. I couldn't do that because I'm the only human. I'm the only human around doing this, but there could be a lab with more than one human who could do a better job. And so I'll write that in more like lofty science sounding words, of course, but
more politically
yes.
correct than
Yeah. But so the idea is small pilot study, somebody else take it and do it better. I'm out.
Okay, perfect.
Yeah.
All right, I will slowly
Yeah.
start going through the questions. Probably rants
Yeah.
will continue. Bear in mind.
Sorry.
Yeah, no worries. I guess I'll go through them in order. That the first question was, how do you quantify? Oh, okay, that there was a word that you said. I'm not very good at this. How do you quantify cell senescence? Senescence
I
in
remember.
your lab subjects. So that was a while back you mentioned it. So sorry, this will be random questions being pulled at different times.
No worries. I remember. So I'll do 2 different answers for that because we hadn't gotten into my specific experiments yet. So for me, for my worms, senescence has multiple meanings. So if you're talking about organismal, like whole body senescence, the term senescence is pretty much just decline with aging. That's pretty much all it means. So, which is different from cellular senescence, which is a super specific thing. But the vague, just like the word senescence, like you could look at your grandma and be like, man, grandma's really experiencing some senescence right now. And like you would be right. Most likely, unless your grandma's like a bodybuilder. I don't know your grandma. But so that's all that it means. And so for my worms, in the beginning, I did experiment with some quote unquote like health span assays. So not strictly just the point that they die, but some other assays to see like how healthy they are. And there are a lot of different ones available for worms. The one that I was using was locomotion. So basically, when I tap the plate or poke them on their little heads, if they move at all, they're scored as not dead. But I tried getting some more information than just not dead, so I would poke them and I would record if they were able to actually like move from point A to point B, like actually like move to a different location, locomote. Like then I'd call them motile, and I scored if they were able to do kind of like a body roll, like a wiggle. And then the ones that were like really struggling, like on their deathbeds on the struggle bus, I would poke them and all they'd be able to do, let me try to extend my neck so I can do that. They'd just kind of be able to go like this, like they'd just kind of like, you'd poke them on the head and they'd be like, I don't like that. You'd be like, and just kind of like shrink their head in. And so I was getting that data for a little while. But I presented it at one of my thesis meetings and we all noted that it wasn't really giving us anything that we weren't getting from lifespan. So in worms, at least for this specific health span assay, it pretty much was the exact same curve just a few days earlier. Like they lost their motility just reliably a few days before they lost their lifespan. So it wasn't giving anything unique and it was taking me like 5 times as long. So they were like, you can just stop doing it. Like it's not worth it right now. But again, in a future lab where they have more resources, I would 100% suggest that they get that locomotion data because it's easy. There are other assays that you can do to measure like the buildup of like metabolic byproducts that they accumulate over time. Another fun thing about worms is they're completely transparent. Like you can see through them, so you can do really cool like fluorescence assays. because even when it's just internal, you don't have to slice them up and section them. You can just look right there and be like, aha, that cell, it's right there. I can see it with my eyeball through this microscope, you know? Even like while they're alive. So you could do some really fancy shit with worms if you have the resources. And then, not to take too long, cellular senescence is a whole different thing. I don't think worms really have that. I don't think they really live long enough for that. Worms are entirely post-mitotic. And so what that means is, all right, humans are not post-mitotic. So you know how like if you scratch your arm, you don't have the scratch forever. Like other cells will come, they will divide
Yes.
and take its place. They do mitosis. You remember mitosis from like 8th grade biology? Worms don't have that. So they have exactly 959 cells. We know where they are, where every single cell has been mapped in the worm. And one, you know, they divide obviously during early development, but once they reach adulthood, those 959 cells are there to stay. No more dividing, no gain, no loss. 959 from the end of worm puberty to death. So they don't really have things like cancer and cellular senescence and all that fancy stuff. But cellular senescence is really cool in humans and more complex organisms. It's basically like zombie cells. They're cells that are kind of fucked up enough, like they should have died, or like become cancer or something, but they didn't become cancer and they didn't die, but they're not doing what they're supposed to do. It's this third option. They're just kind of hanging out and producing like inflammation and messing with your body. We think that those pathways exist because they're important during early development and during wound healing. But they're supposed to be able to be cleared by the immune system once it's time. So like acute little senescence cells are okay, but when they become chronic, they just kind of cause trouble. So there's actually like a lot of interest in human research in figuring out how to get a handle on those senescent cells, and use anti-senescent cell drugs as anti-aging therapies. But that's a whole other can of worms, and I'm gonna let you go to the next question.
Can of worms. I like. No, that's a very good question. I wouldn't even thought of asking a question. So it's a very good one from Science Fish. I suppose this was asked at the beginning of the interview when your camera was set up initially. But someone was asking what game is in your N64 right now.
Oh yeah, I think so. That's my partner Jason's desk. Yeah, so he has that, and he actually, I want to show this off because he's very passionate about it. He also has his little like matching. My little noodle arms. Can you see? There's a little matching mounted controller too, and it's Super Mario 64.
Super Mario 64. There we go. I moved the camera so everyone can see.
Oh, sorry. Yeah.
No worries.
Yeah,
no worries.
it has the matching little controller too.
Nice. Oh, that's brilliant. The Nintendo 64 is really, really good. Very good question. Off topic completely, but very good question.
Yeah.
Another question. Again, this was a while back, so keep your brain. Don't be confused. Right. It was a while ago.
I won't be confused.
What rough scale of lifespan change have you affected with experimentation? So I suppose you talked about the results and you found the genes, but by how much was it prolonged? Yeah. What were the results?
It's a really good question. So the quantity of lifespan extension is very dependent not only on the specific genes that you're affecting, but also on the method that you use to impact them. And I was using a very mild method for complicated reasons that I can explain if you want. But let me say that the quantity of lifespan extension that has been achieved through genetic modification in worms is much higher than what I'm seeing with my methods in my experiments. So with like the fancier methods, you can get them to live 8 times as long. Like it'd be the human equivalent of like 800 years when you're actually going in and doing a what's called like a germline mutation and like altering the actual sequence of their DNA from birth. Now, I didn't want to do that for 2 reasons. One, it's hard. And like we've said, I didn't have the money or the time or the people for hard things. But 2, It is those kind of mutations are not what I want to study. I want to study interventions that are happening specifically in later life. And the reason for that is because of a concept of antagonistic pleiotropy. And I can break this down pretty easily. So pleiotropy just means one gene having multiple effects. So pleio, multi, tropy, target, like multiple targets.
Okay.
So one gene, multiple effects. And antagonistic pleiotropy just means one gene, different effects that oppose each other. And so there's a very big concept in aging, the evolutionary theory of aging, where we think that a lot of the genes that are very important for early development, like, embryonic development or early childhood development, puberty, anything up to and including pumping out as many babies as possible, because that's what evolution wants. That many genes important for that might actually have negative effects later in life.
Yeah.
Because evolution doesn't give two shits about you once you get your babies out the door. Because the way natural selection works is, I mean, if you remember from basic evolutionary biology, the idea is that any mutation that shows up that allows you to have more kids and raise them to the point they can survive on their own and they can go have more kids, that's going to be naturally selected for, because those genes are going to survive, because we just said you have children, they go forth. After those children make the next generation, you are completely irrelevant from the standpoint of evolution.
Yeah.
Evolution, like, you can go ahead and die, you can suffer. Evolution doesn't care. Evolution is a cold bitch that way. So once you have your babies, it's done. And so there are many situations where there could be a mutation that lets you have like double the amount of babies, but then as soon as you hit 40 years old, you get dementia or something. Evolution would look at that and be like, yes, ship it. We love it. Evolution loves that. We, on the other hand, might be like, hold on, I'm actually fine not having 10 babies and would prefer not getting dementia. Can we walk that one back, please? We might have made different decisions if we had a say in it. So that's the idea of antagonistic pleiotropy. And because of that, I didn't want to do germline, like day one mutations in my worms, because my theory is that a lot of the genes that might be important for aging, if you mess with them from day one of the embryo, the worms might just die, because it might be really important for embryonic development, early development, or having offspring. So I wanted to specifically only mess with them once they reached young adulthood. And so this is a much more mild intervention. And especially because I wasn't doing it the fancy way by changing their DNA and using a chemical to induce the change in a time-dependent manner. You can do that. It would have taken a lot longer and been a lot more expensive. I'm doing it in this really fun and easy way that was invented in like 1988. It's not fancy, but it works so well. And basically, you can raise up bacteria that produce what's called RNAi, RNA interference. It's these little chemicals that will go and bind to the DNA that you want to turn off. And suppress it. And so you can just grow these bacteria. It's super cheap. You just just in little bacteria culture, little antibiotics, so easy. Grow them, add this one chemical that helps them produce the RNAi for your specific gene, and then you literally just pour that on the worms. And the worms eat bacteria. So then they eat the bacteria, and the bacteria have all the RNAi in them, and then boom, whole body RNAi. Then you're suppressing that gene in the worm's whole body, and they can just do it with food. If you're not in the lab, you might not realize how amazing that is, but normally it is much more effort to do whole body RNAi. You can't just like feed it to mice. That won't work at all. They will just digest it, and nothing will happen if you just feed it to them. Worms are special, and it makes doing like huge RNAi screens very accessible. and it's just amazing. So anyway, answer the question, I get 10 to 20% lifespan increase. It's not like mind-blowing, but for the extremely mild method of literally just feeding them some weird bacteria, it works, and it only needs to be detectable. I just need yes or no. I'm not trying to break new ground. This lifespan extension ever. It's not the point of my study. My study is just yes or no, and it was enough for yes or no, and so I'm fine. Yeah.
No, I mean, preserved digital sounds mind blowing to me. It sounds really good to me as well. I mean, consistently, and again, like you said, reproducibility. Reproducibility is really important. Although, I suppose the big talk about reproducibility is that you might not get to know that because publication of reproducing results is tough to get there. So as long as you publish your papers first, might be tough to ever see anyone else do it as well. So that's the downside of it, unfortunately. I will send you the other question. It was a really long question.
Okay.
And I'm not. I'm sending you to in Discord just so you can have a read of it because it's a long one. But I can read it out for chat. But just bear in mind that it's a long question. Telomeres are part of a chromosome that protects it from becoming damaged or unusable as it condensed after DNA replication. They shorten with each cycle, eventually depleting, essentially being the limiting factor to natural human lifespan. Can you talk to us about the state of research in this field and how close are scientists to finding a solution to extending human life through this means? Or correct me if I'm wrong.
It's a great question. Okay, so yes, telomeres are the parts of the chromosome at the end, and the basic idea is the way that DNA polymerase has to bind to DNA in order to make copies of it. It can't bind from like the tippy top of the end. It kind of has to like grab on. So it can't grab on up here. It kind of has to grab on like a little bit of the ways down. So everything at the top, it kind of just skips that. So it's nice having telomeres, which there's just kind of this series of repeat of like random junk that you don't need at the end, so that when it can't handle the very tippy top, there's nothing important there. But each time you do that with each cell division, it's going to get shorter and shorter. And the theory was that if you keep going long enough, then eventually you'll start to hit important stuff because the telomeres will run out. The thing is, there are enzymes called telomerases, which can add some back on. And telomeres were really hot at a certain point in time, maybe like 10 years ago, but they've become less hot. The research has shown that there isn't a strong correlation between the length of the like natural length of telomeres and the lifespan of the animal. For example, like mice have super long telomeres, but they still die after 3 years. So it doesn't seem to be the big main driver of lifespan, at least as current. That's my current understanding. I mean, science, there's give and take. Some things, some theories like get popular and then unpopular and then might have a resurgence later. So take it with a grain of salt because I don't specifically study telomeres. But yeah, my sense of the current research landscape is that it's not like number one priority right now.
Okay. So there's other focuses right now for longevity of mankind or human species.
So we don't think it's like the limiting factor.
Okay.
And for worms, it's completely irrelevant because their cells don't divide at all.
Yeah.
So I can't study it.
Telomeres are so out of style.
Yeah, not hot anymore.
Another question is, did you study reversing aging as well, or is that a fairly different topic? Did you touch on reverse aging in any way?
So it's interesting.
Is that a real thing, or is that sci-fi?
I think it's just a matter of it can come down a little bit to semantics and philosophy as well as science. So it depends on what you mean. And even getting people to agree on a definition of aging it can be difficult. And for example, lifespan is kind of the golden standard for aging research because it's the one thing everyone can agree on. It's pretty cut and dry. You either dead or not dead. But other proposed markers of like how well you're aging, so to speak, or how like internally young you are, like your so-called biological age, might be getting like Instagram ads for like, oh, measure your
I've
biological
seen
age.
that so many
Yeah.
times.
Yeah. So those are controversial. That's not like established science.
And it's funny you say
It
that
like
because it always says scientists say at the top always.
yeah, I mean, I'm sure there are a handful of scientists that do say, but for every handful, there's a couple handful of scientists that don't say.
Mm.
And you're gonna come down now to personal opinions. So I have personal opinions on a lot of those as well. So a big one is like epigenetic clocks. And this might be like a little bit of a rabbit hole. I'll keep it short, and then just if people have follow-up questions, we can come back to it. But basically, epigenetic means like on top of your genes. So you have your gene sequence, but then
Yes.
you have all these little modifications on top of your gene sequence that kind of affect how much those genes actually get used on a daily basis, basically. And so epigenetic clocks are looking at those little bumps and squiggles that end up on your DNA. They change over time, and there's some research to suggest that measuring like the presence or absence of some of those key little bumps on your DNA could be a marker for like a good way to measure like your internal, like your biological age. But the thing is, for me, I'm not a huge fan. I've seen a lot of conflicting results, and for me, I'm very big on correlation does not equal causation.
Of course.
So like just because you can go and maybe take some do something and change the bumps on your DNA, does that really mean that that's going to correspond to like better heart health? I don't know. It's hard to prove. People are trying, and I think people are making progress, but it's a very nuanced topic. It's not cut and dry. It's not a done deal. I would say like the science is real, but early. So I would take those things with a grain of salt. Like if you want to do one of those Instagram ads and you're like, I have disposable income, this looks fun, then fine. But don't do it and then panic if it says your biological age is like 95. Just be like, well, shit, that wasn't as fun as I thought it was, but it's probably bullshit. Just don't panic is my point. Yeah.
Okay.
Yeah.
That's
Did that answer the question?
That was the reverse
Forgot.
aging. Yeah, no, I had to look back at the question. But it's okay.
Shit.
We got sidetracked, but it was a good sidetrack. I like that one. That was good.
Yeah. Wait. Sorry. Let me just say one more thing. Then try to stay on topic. So like there is research to try to like, oh, let me go back on like that epigenetic clock or pick another marker and like try to go back on that. But for me, I don't care personally unless it's like a very functional outcome measure. So can we reverse aging in terms of like improve your heart health or improve like frailty? And then if you're looking at something like frailty, like how strong you are or how likely you are to have fractures in your bones or something, then technically we already know how to reverse that aging. It's exercise, and then maybe taking like vitamin D and calcium if you're a postmenopausal woman, and also weight-bearing exercise to improve your bone health. Like that stuff doesn't sound sexy, but technically if you want to reverse aging in terms of functional outcome measures like frailty, it's going to be things like that are the most proven right now. It's just like be healthier.
Nice.
So yeah.
No, I'm glad you said that because I went to the gym today and I got my vitamins. Didn't have protein today though, so that's a bad thing.
Nice.
But yeah.
Yeah.
Okay, so I guess that's the last question we have. So I have
Really?
tracked a few of my own. Damn it, Durap just ruined everything. Would these RNA antagonists be able to change genes in gametes?
So like the RNAi, I guess, but it doesn't really make sense because if you were going okay, hold on, I might have misunderstood the question.
Oh, sorry. Do you want me to say it or?
No, it wasn't your fault. It was me interpreting it differently. So do you mean like if I throw my RNAi at an adult worm who is pregnant, for lack of a better word, they don't really get pregnant, but you know what I mean, who has eggs, and they're gonna lay the eggs, will the RNAi affect their offspring? Um, mm, it depends. Not in a very predictable way. Because it's very transient. And like you can you can there are things you can do that will affect the next generation. And it's not even just genetic interventions. Like if you starve the shit out of your worms, the next couple generations of worms are going to be a little funky. Like they will sort of like remember that in their genes that they went through starvation. And that's been something there's been research on that in humans too.
Oh,
That the
yes.
physiology of like the grandchildren of people who went through like severe famine have detectable changes. So what there are things you can do to creatures that will affect their offspring, but in terms of like the affected like directly modifying the genes of gametes with RNAi, we wouldn't really do that. Like if we were trying to do something from not even just from birth, from pre-birth, we would just go ahead and do a mutation because it's permanent. Remember the reason I didn't do a permanent mutation is because I wanted something only in adulthood. Does that hopefully that kind of makes sense?
Mm. Yeah, no, that's very good explanation. I understood. Lint is asking, do you know about the open worm project? This was brought up during science stream, actually. Do you
Oh
use it
yeah.
in your research? Is the question.
I haven't been using it in my research because I am my research is pretty simple. Like I said, it's pretty binary. I just like throw the bacteria food on them and I poke them and I write alive or dead. So I don't really need a lot of super fancy information for that specific project. But I do know about it. It is super cool. I just like opened the website for reference now. OpenWorm is an open source project dedicated to creating the first virtual organism in a computer. Because modeling a simple nervous system is the first step towards fully understanding complex systems like the brain. And yeah, so I haven't used it, so I don't want to like talk out of my butt about it too much, but I will just say, in general, worms are really cool. Like I said, they're see-through. We know exactly every single 959 of their cells, what they do, how they map to each other. So that includes neurons. So we have like the entire cell-by-cell nervous system all mapped. We can see it in real time because they're transparent if we add little like fluorescent baubles. It's actually the first ever multicellular organism to have its complete genome sequenced. And yeah, worms are super awesome. And there's all these like worm specific materials. There's Open Worm Project, there's Worm Book, there's Worm Atlas. The worm community is like very passionate and has lots of really awesome like open science tools to share with each other. So I highly recommend it as a science community. Worms forever.
Linking from that, you talking about worms, that was actually funny because it reminds me of my question towards science actually when he was here. So I was kind of gonna say, like, I know you know the memes of biologists hate, chemists hate, physicists. That there is really no hate. There's more specific hate. For example, physicists and engineers is the big joke constantly, right?
Yeah.
And then I asked science if there was, and he said that there was this discrepancy between ant people, worm people, and there was another classification. I forget the third
Flies,
one.
maybe?
Maybe flies. So there was like these three groups and three cliques, and you all like don't get along with each other. My question is, in your experience in throughout your PhD in this field, is there cliques? Is there like a group that hates the other group, or is there any drama with other departments? Obviously,
There's
don't have
definitely drama.
Okay.
But I haven't seen like worm versus mouse versus fly drama. The drama like that I've mostly seen is just like researchers getting opposite results in the same field and then shitting on each other.
Oh, wow.
I would say the big like organism based drama, it's not even really drama. It's just sometimes, especially as like I'm supposed to be a medical scientist, right? So my research is supposed to have applications for human medicine. And so for people in medical departments like mine, sometimes when you send out your manuscripts and it's worm based to biomedical journals, they are very skeptical that it's going to be interesting and relevant to their readership, because they're like, we want mammals, like we don't believe that worm, they might not be very familiar with it. It can be maybe hard to get grants. Similarly, for worm research, if you're reaching out to like medical granting agencies, because again, they're skeptical that it can be relevant, and so that can cause some tension and annoyance. I'm hoping that I saw that coming and headed off at the pass with my research, because all the data that my study started with before we went to worms, all those 25 data sets that I did my meta-analysis on to find my candidate genes, that was all mammal data. I had data from mice, from rats, dogs, humans, lots of human data. So I have baked into my manuscript, and I'm trying to make this really clear in the introduction and cover letter, like I have established human relevance before I even started the worm experiment. So yes, it's worms, but like
you're not going in blind.
Every single candidate was already something that in like a totally separate part of my experiment, I flagged as super relevant to mammalian aging. So it's not guesswork. We don't have to wonder if it'll be relevant to mammals. Like I already slid that in there at the beginning. So I'm hoping to like head off that challenge at the pass. Yeah.
Okay. So, but I suppose to go back then there isn't too much drama as much as like I kind of questioned that, rather it's the whole medical aspect rather than fields of PhD research that
it might depend on so the thing is, like, I don't study worms for the sake of studying worms. They're a model for biomedical research. So maybe someone like science has sort of a different experience because he's like studying ants. Like he's like, the whole point is to find out more about ants. And that's kind of like a very separate segment of the scientific community. Like I'm in a very medical area, so the sources of drama might just be very different. And so maybe in his area, it's people being like, well, why do you want to study ants? Like studying flies is so much cooler, Whereas in my area, it's more like, nobody really cares about any of them. We just want to know what's the best model for humans.
Fair enough. Yeah, no, absolutely. Fair enough. Anyways, it was just a bit of a banter, lighthearted question. Because I always want to break down the bridges that, people are burning. Yeah. Wait, why do I want to break down the burning bridges? That actually doesn't make sense. Anyways,
build the bridges.
Yes, rebuild the burned down bridges. There we go. Does AI play a role in your research somehow? How do you think AI will affect those researchers in future?
That's a great question. So my project got started pretty much like before the advent of AI. So it didn't play a role in the design of my experiment, but so how, like I said, my labs just like evaporated while I was here, so I've been all on my own. So my instead of having like a real PI and a bench mentor and like friends in lab, I have ChatGPT. So when I have a problem that comes up in my research, I'm like, ChatGPT, I don't know what happened. Like, I don't understand. Like, did this antibody didn't work? Like, what do you think is going on? And it's so supportive. It's like, oh, that can be really frustrating. This is actually a common issue faced by scientists. And one approach that we've seen a lot of online, is blah blah blah. And like, here's a great reference.
Budget therapy.
Oh my god, it's my therapist, my bench mentor, my like substitute PI, like my postdoc, my best friend, my lawyer, my accountant, like really just saving my butt. And I wish I had ChatGPT when I was doing the computational part of my experiment, because coding is its native language. It has to translate code to English so it can talk to us. And so when I go in now and I want to go back and change something in my code, Like I can just copy paste it into ChatGPT and be like, hey, I don't remember what I wrote here because it was 3 years ago. Can you help me with this? Like, I want to change this to like select the best 5 instead of the best 10. I don't even remember what line that was. Like, can you figure this out for me? And it'll be like, yeah, and actually, like, here's a more efficient way. So I don't know. People are gonna have a way easier time doing research now they have ChatGPT, especially computational research. Like, it's just completely changing the game. But in terms of like future studies, I would say it's hard to predict exactly what'll happen, but I think things are really gonna change. Like I think AI is going to be able to propose the most logical next experiments, like write its own grants pretty soon. Um
Yeah.
it's going to be very hard to find like a human written grant or paper in the future because I think the way that technical writing works, like grants and scientific papers, I think that's going to be ChatGPT's bread and butter. It's going to do a
Absolutely.
better job than we can. And literature reviews, I think some people have already tried this. So literature reviews are when basically like you try to read every single paper on a specific topic and then synthesize the results. And back in my day, I had to actually go and read all the papers and think with my human brain and write a summary and do analysis to find patterns. Now we might be able to just write a prompt in ChatGPT and be like, can you read all the papers ever written about the RNAi feeding method in worms and tell me the 5 most interesting things you think you learned from that? And then it'll probably just tell me and I'll be like, great, write that in the style of like a literature review published in Nature. And then I think it just will. So it's really going to change things.
No, that's really interesting. And like especially Volcano Doc coming in, welcome in, mentioned this. The way you talked about the code was actually ingenious because I should have freaking done that because my first few years of my PhD was all simulation based and programming
Yeah.
based. And then like I didn't end up putting it into my thesis, but I wanted to put it into what's the called? The appendices. I just wanted to add it in that I
Yeah.
did all this research on
Yeah.
the electric grid and on battery
Yeah.
simulation. But I was like, oh, this is like 3,000 lines of code. I don't remember any of
Yeah.
this. I don't want to go
Yeah.
through this. And
Exactly. I don't
I
remember
should
what
have,
I did anymore.
yeah, I should have done what you said. That was really good.
Yeah. Oh wait, I
Um,
see Volcano Dot disagreeing with me. And wait, I have a quick follow-up
okay.
question. So you said you've convinced it of stuff that's not accurate. Yeah, I mean, if you definitely have to know how to talk to it. But also, did you use which version did you use? Because I definitely saw funky shit with 3.5. 3.5 can't do math. It's tough. But 4 and above, now they have 4.0. 4, I haven't had like any issues with. It's
Mm,
incredible. And I know what I'm talking about. I would know for my field at least. And if it
mm,
said something wrong, I mean, it depends on like the body of, the quality of the data it has available. So maybe the quality of the data it has available for some reason on volcanoes is shitty. I don't know why, but it's possible. Certainly, I'm not denying your experience, but I'm just curious which version you talked to, because I've had great experiences. And
mm.
it might just be a matter of prompt engineering and knowing when to second guess
Yeah. No,
something.
prompt
But
engineering is very important.
yeah, I mean, but I've just, and I always double check. If it says something that I don't know already and it's not just like reminding me of, I always go and like check it. But it's much easier to go check for something that I already have the information in front of me. Like I can just Google this specific outcome that it told me than trying to think from scratch of like what to look up when I'm first trying to figure something out. I don't know. It's hard to explain, but
Yeah.
I find it incredibly helpful as long as you're like careful, just, be reasonable with it.
No, no, I had basically a coin kind of experience. I had same kind of issue. One, when I was preparing for my defense, I actually did use ChatGPT for my defense. I probed it on thermodynamics, on Gibbs energy, and chemical potential, things like that, because obviously you do that in your first few years of physics, and it's been
Yeah.
8 years since I did that. So
Yeah.
I was like making sure that for my defense, I was prepared on the foundational fields of thermodynamics. and I was using ChatGPT a lot, and ChatGPT did help me because I kept probing, saying,
Yeah.
oh, what about this way, what about that way?
Yeah.
But obviously the other aspect of it is anytime I looked up anything specific about vanadium flow batteries, it would bullshit recite my papers. There's only so many papers out there.
That's funny.
In vanadium flow batteries. There's like three different we'll say three biggest researchers out there, and one of my research group is one of the bigger research groups of it, and it literally recited an abstract I wrote before in the past. I was like, for frick's sakes, this is useless, this is not new information.
Yeah, I mean, C. elegans research is nice, especially maybe my experiences are different because, like I said, due to like sheer necessity, I'm using very classic, read old and cheap methods.
Yeah.
Like most of the stuff I've been doing has been around for 3 decades, so ChatGPT knows its shit. Yeah.
No, that's completely understandable. Perfect. And to move on from AI, I have one more question here. What is the most accurately simulated organism so far? Talking in relation to the open source you mentioned earlier, the open worm, I think it was called. And are they functionally so close? Oh, sorry.
Oh, sorry. I was just clearing my throat. I still have some COVID in there. Go ahead.
And are they functionally so close so that scientists are able to make any meaning in inferences? Yeah.
I might have to just give an I don't know for this one. Because that's not really something that I've looked into directly. I think it's a great question, and it's something like I would love to spend a stream just like Googling that together and trying to learn more about it. And I might be able to make some more, educated guess conclusions based on what we're looking at and be able to sort of like guide the science reading. So if that's something that you want to explore during a stream or like post about in my Discord and we can talk about it there, async, I would love to do that, but I would just be making shit up if I tried to answer without doing any research. Yeah.
Perfect. I suppose there was a question there following from AI, if there was any good AI that you suggest for uploading PD files like books and papers, etc. But Slightly Caffeinated actually just jumped in here to say Research Rabbit. I guess we have a few researchers in the chat as well. What program did you use? So I used what's that one called? Medley. That's all I used to keep track of citations. But I unfortunately wrote my whole thesis without the help of ChatGPT. I tried. I'm going to be honest. I tried. I never ended up doing a single line of ChatGPT or help. So if you used any AI to help you in any way, or to keep track of papers or research, did you use anything?
Me or
yeah. No, sorry. Yourself.
I haven't used AI to keep track of my sources. Like like I said, I started this before AI, so I had like a citation management system before I even started. I actually use a little like a more obscure tool. Has anybody heard of Citavi? Citavi? Have you heard of this, Tofu?
No.
Yeah, so I really like it. It's definitely like doesn't have the same like, bid team and following as Zotero or Mendeley or anything, but I'm trying to figure out how to explain it. I'm gonna put the name in chat if anyone wants to look it up. I'm a citation hipster. Does they have like a little intro video that'll explain it? But basically, like you can add little like you can organize an outline of your paper like in a panel in the app, and like in all the references like add different like highlighted regions and notes and memos and even like screenshot little like figures, and then you can like drag and drop all these little snippets you have from each paper and thoughts you have tied to specific parts of the paper into your outline of your paper. And so then at the end of it, you kind of have an outline that's just made up of all your little quotes and thoughts from what you're reading in order, and then you can just sit there and write it, and there's a plugin for Word, and so you can just like in order read like these are all my thoughts, and then you click the button to like add the citation for that thought, and it's like already written there in an outline. And I'm like very anal about like wanting to make sure like everything is in order
Sorted.
before I start writing. So I love it. And also, my thesis proposal hasn't really changed very much since I wrote it to now. So I'm actually able to go back, and like the outline that I had for my thesis proposal is like very similar to what my final paper is going to be. And I have very little recollection of what I was thinking when I wrote my proposal and where I got all those references 5 years ago. But now I can just scroll through and it's like, oh, where did I learn about the RNAi feeding method? And it's like, I go like thesis proposal, methods, worm methods, RNAi feeding, and then it's like all my thoughts on like my like 5 thoughts on that, like with their papers tagged, like right there. And like I just love having it organized like that because I don't trust myself to remember anything, and now I don't have to. Yeah, so I love it.
No, no, that sounds absolutely fantastic. So hopefully, a few people are picking up some of these things if they need it. But as always, by the time you start searching for a new method of keeping track of everything, it's too late. It meant you haven't been keeping track, and it's all over now. I found that was my situation writing up my thesis. I left everything to the last minute. I was very bold, and it was a mess. Oh well. Oh well. But that was officially all of the questions asked for and covered. I will ask one final question, if that's all right with you.
Okay. Yeah.
And I will basically let you finish up in any way you'd like. But my question is just a bit lighthearted, because this is something that's come up several times as an expert in your field, a researcher in your field, about to get your first doctorate title. Would you be called yourself doctor? Anyways, side joke.
Yeah.
My question is there any good movies out there or TV shows that have, in your opinion, have good science based on like your field? So obviously, like I could talk about the physics of like, I don't know, some movies have some physics talks in the background, and I usually complain mostly then say there's good ones. But do you have any comments of some like movies that are absolutely fantastic or absolutely terrible out there?
Ooh.
I guess in a lab biology setting, I would assume the question would be aimed at.
I'm gonna ramble a little and try to let my brain try to think of something specific on the back burner by the time I get to the end of my ramble. But because I'm not sure, I'm pretty bad at that. Like my mind instantly goes blank. But I will just say, like in my memory, every time I watch any show that has wet lab research, like biology research, I always am shaking my head. It's pretty much always terrible. And I think the reason is that, like, in real life, like anything you're doing in wet lab, like if they did it realistically, the movie would be so boring because they'd be like, oh, like we need to figure out like what this mutation does. And the reality would be like, let me write a grant, and then in 6 months, we might know if we have funding for this, and then we'll start planning our experiment next year, and then it'll probably take 2 more years from then, and we'll but in the show they're always like looking, and they're like, I see the DNA right there in the microscope and it changed, and I'm just like, like, no, you did not see the DNA in the microscope. And like, what, you just had all those reagents lying around, and I don't know. And there's always like multicolored liquids. Like, that's not a thing in biology, like maybe chemistry, but it's just, if anything is like glowing or multicolored or exploding and you're in a biology lab, like you have done something very wrong, and you should probably call emergency services and leave the room. you know so like I think real wet lab science is just way too boring for movies and slow.
Absolutely understandable. No, I can understand that from lab practices. So there has never been a movie where you were like, oh, that's really good science.
Ooh, I not that I recall, but that might be something that I have to like mull on. And then I'm in chat. Like I'll be in chat another time. And so if I think of something, like I'll bring it to you in chat on a follow-up stream. if that's okay.
No problem. Of course, of course. No, that's perfect. I suppose my final kind of sentiment or anything is that, you know, maybe you prepared to say something and we didn't get a chance in the end. If you have any personal commentary that you'd like to add, your drawings as well, which is absolutely amazing. And of course, you sent me chunky rats, and yeah, you guys should check it out because she has chunky rats in there.
Chunky rats.
No, no, it's absolutely. And is that so? Is that all you do at the moment on Twitch? You stream
No.
drawings. What else do you do on Twitch then?
Hmm. Yeah, no, I have trouble doing just one thing. I have too many hobbies. I like trying lots of different things. Um One thing that I got super into because I joined Twitch and was exploring like the art and makers and crafting spaces is I got really into mechanical keyboards randomly. I stumbled upon a keyboard streamer early on around the same time I found you. And I just loved it. Wait, am I frozen or I
No,
don't know,
no,
just
all
let me
good,
know
all
if
good.
okay. Okay. So I loved it and now I like build mechanical keyboards and I also started making my own custom keycaps, like artisan keycaps.
Oh,
I have little like it's like tiny origami in resin is one of the styles I've been
oh my
doing recently and
days!
I don't know, it's pretty hard to like
Get
see,
in
but
focus. Do you have
yeah,
an
but
Etsy
like
or something?
I have a shop. I just started the shop. Like I
Okay.
like just like so there's just like a couple pre-order slots and like I'm still like kind of learning the ropes. I just finally got a PO box last week so that I can do shipments without just like telling the entire world my apartment number. So it's like very early days, but that also means everything's cheaper because it's still just prototypes. So like get it while it's cheap. My shop can I put my shop link in if
Absolutely,
anybody wants to
absolutely.
see? Yeah, so my shop link, it has, oh, and I can show you more keys if you want. Oh, and for people who don't have mechanical
I'm just gonna
keyboards, some
open it.
people still liked the origami. So I started making like pins and keychains too. So like, hold on, this camera is like not the best at focusing on tiny things. But like there's a tiny origami in there. It probably won't. It probably will never focus, but like it's in there. You can see the pictures on the website
Yeah, I'm looking
better.
at the website.
Uh,
I see the
and so
exact one
like
you're showing.
it's like a little pin, and if I just grab like one thing from up there. Oh, but also look at this super cute keyboard. So I have like really cute keyboards now. Like this one has cat ears,
Matching
and
your headset.
yeah, and I did my nails to match the one that I'm using right now. I have so many keyboards now. Hold on, let me just get the little ear lights to turn on. See the little ears glow and my nails match.
Nice.
I got little
This explains
paws
a
on
lot
it.
about your stream now. Every time I've caught it, there's a camera
Yeah,
at the keyboard. Okay, yeah, no, I get it now. I'm a
yeah,
bit delayed to the party, but I get it now.
I'm in my mechanical keyboard phase. I know it's random and it's like not science, but I enjoy
Oh
it.
no.
Um And yeah, hold on. Let me just grab one thing. Oh, and Mochi came in and is meowing. So the other element of my stream is my co-streamer Kat, which I can show you, and she is featured on my website too. But like, here's a few. I just sent out some, so it's kind of empty. But like, there's a few more in there. You can see
They're
all these
absolutely
little
beautiful.
crane keycaps. And I started making. There's like a meme. I don't know if there's any way for me to explain this normally, but I maybe just won't even try. I'll just show you the keycap and let you think what you want to think. But it's a little booby. It's a little
I was
booby
going to
keycap.
comment. I saw that, but I wasn't going to say it.
Yeah, it's a little booby keycap.
It's called
It
Tuesday.
even yeah, there's my stream team is the Moco Loco stream team. It's like makers and
Ah,
crafters,
yes,
small
yes.
artists and businesses. And on Tuesdays, they have like Titty Tuesdays, and Derek wears these little nipple tassels and like spins them. I don't know. It's normal there. It's kind of like you had to be there. And so I launched my shop last month for they had their big annual like partner party, which they use to promote small businesses in the Twitch community, like maker and crafters business, and I wanted to participate, so I gave away some keycaps, and I wanted to make some special stuff, so I made little booby keycaps, and I made
That's really
keycaps that
nice.
have like their Moco Loco logo on them. Yeah. And then Mochi actually just graced us with her presence, so I could show you my co-streamer potentially. Come here, baby.
Hello!
Yeah,
You're
who's
so
that belly?
cute.
Where's my baby girl?
Look at that kitty.
And we have redeems on stream to give her like treats and catnip.
Uh-oh.
One time we gave her so much catnip that she rolled over on her perch and just completely fell off. She like got too high. It was pretty funny. There's a great clip of that. Yeah. We have another clip where she punched me in the face and actually had really good follow through. It wasn't a bop. Like she punched with follow through in my nose. Yeah.
That's how she shows love, clearly.
Yes, that's how she shows love. Yeah, I just thought you might want to see her and her little belly.
Absolutely. Absolutely. We're very
Yeah.
delighted to see lots of cats emotes and wows emotes in the chat.
Yeah.
Tofu, our cat's
As
fluid.
the other thing that I do on stream, I'm like trying to remember. I'm such a chaos goblin. Lately, just because I need to do so much writing, I need to write my manuscript, I need to write my dissertation.
Oh, you're doing the study thing.
Yes, I'm finally living up to my name, AriellaStudies, and we're doing co-working streams. And I try to make them fun. You know, we have buttons to like, you can make me change my keyboard to see a different keyboard. You can obviously give Mochi cat trees. We have like a joint task list. So you can add things to the list and cross it off, and we can celebrate when you get something done. And then we'll have little breaks and plan something fun for the breaks. You know, we do the Pomodoro timer, all kinds of stuff.
Yeah,
So,
this is a
you
really
know,
good
if you want
method.
to get work done with me and help me write my PhD without being super miserable, I would love to have you there.
I should have done that. That would have been a good tactic. Although I'm not very good at multitasking. I just
Yeah, it
never get
is
anything done,
pretty hard.
unfortunately.
Yeah. Yeah. Oh, there's so much cat fur on my face now. Oh god.
And you still have a little bit of COVID left. No.
Yeah. Yeah, my voice isn't usually, it has like a slight husk to it. I kind of like it. It's kind of sexy, but yeah, I don't normally have that.
That Phoebe episode from Friends when she had the husky voice.
Yeah.
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 Well, yeah, Erin, thank you so much for taking time out of your day to come on to Tofu Tech Talk. I really appreciate it. I've been reaching out to many people with STEM backgrounds, but also it's just really nice that you were so flexible and got everything ready and was so excited. It was honestly such an honor to have you on the guest as a guest. You were great banter as well, and good crack is what we would say here in Ireland. It doesn't mean what you might think it means, I promise. That's my
I was
personal
so
commentary.
excited. Yeah, I was so honored. Like I said, you're one of the first I ever followed, and I was like admired and like intimidated by you, and so it's like a dream come true to be here today.
I hope I wasn't intimidating. I hope I don't come across intimidating
Not
most of
on
the time.
purpose. No, just like, oh, like look at you, like big streamer, like so many followers, like so confident, like that kind of thing, you know? Not like mean or anything.
Fake it till you
Yeah.
make it. I wish I was confident, but I really
Yeah.
appreciate the compliments. But I hope anyone in the chat doesn't get intimidated by me ever, because as you guys know, I'm a sensitive snowflake and I'm not scary at all
Oh,
when you talk to me. Yeah.
that's
But
cute.
again, really appreciate you being on the guest. It was really insightful. We'll talk a little bit off stream, but if you'd like to say your farewells,
Yes.
I will go into the AFK screen.
Sure. Yeah, oh, and somebody commented on my Catsmopolitan shirt. Yeah, this is in my shop. Yeah. Calico is the new black. It's Mochi's face. It says flaunt your toe beans, secrets from the litter bots, and 7 tips to open any cabinet.
Brilliant.
Yeah.
All right.
Yeah.
Tofu intimidating. Exactly.
Okay.
Mm-hmm.
All right. Yeah. Thank you so
Thank you
much.
so much, Aurelia. Farewell. And if you guys don't follow her, make sure to do her link. We gave a shout out earlier. We can give another shout out again. But also in the title, if you hover, you get all the socials linked immediately. And yeah, I shall see you guys on Friday for Physics Friday. Take care of yourselves. Bye.
Thank you so much.
I'll
Bye.
show it to you in a second. It's completely balanced.